Great.
So yeah, my name is Kathleen. Like Frank said, I currently work at Fauna Bio. I did my PhD and postdoc work at UCSF and the Gladstone Institute. So I'm going to start off by talking about some of my academic work, and then I'll also give you a tour of Fauna Bio. So the title of this talk, as you can see, is Genome Evolution over Multiple Timescales and Dimensions. And you might have guessed from some of the pictures that here I'll be talking about some things that make us uniquely human, as well as how the 3D genome is involved in this, where this image is the 3D genome. These different colors represent the chromosomes. This is it folded into the small space of the nucleus, and showing how the different chromosomes stick together, essentially. And then another focus of my talk is HARs, or human accelerated regions. And what these are regions of the genome that are highly similar or the same in most species, but then in humans specifically, they've acquired many more mutations than expected by chance. And so this indicates that they're evolving very fast in humans specifically. And so based on that, they may underlie some of the things that make us uniquely human. One thing that's always been a little bit mysterious about HARs is that most of them are non-coding. And so you can see here that many of them occur in introns, many of them are energetic, some of them are predicted to function as link RNAs, and then some are predicted to function as pseudogenes. But overall, they're not genes, so it's a little tougher to figure out what they're doing. However, as we've heard a lot already in this workshop, the genome is much more than the 2% that encodes genes. And so this includes elements like enhancers, promoters, different non-coding elements that drive the level and the environment in which genes are expressed. There are also things called insulators, which can separate different genomic environments from each other. And there are silencers, which are basically the opposite of enhancers. They turn genes off. And enhancers, in particular, are non-coding genomic elements that control gene expression, and these can have important phenotypic impacts. So in this paper, they looked at an enhancer of the gene sonic hedgehog, which is known to have an important role in limb development of your fingers. And so we know that this enhancer drives expression of this gene. We can see this using reporter assays where the enhancer drives expression of a reporter gene, and you can stain for that and then see where the enhancer drives expression. And misregulation of this enhancer or this gene can lead to different polydactylies, as seen in this very cute cat. And so in this paper, they were looking at which parts of this enhancer are important for these different phenotypes that we see, these polydactylies. And so you can see that by doing these different types of mutagenesis, they were able to identify different mutations that would reduce the expression of this gene based on manipulating this enhancer. You can also identify mutations that cause loss of expression of this gene. And you can also cause gain and loss ectopically by manipulating this enhancer, as well as normal expression and reduced expression. All this to say that enhancers are important. And enhancers can be predicted using characteristic epigenomic marks. So as we've heard a lot about in this workshop, epigenomic marks are important. They allow us to interpret different parts of our genome. And one way that we can do this is by using machine learning models, which take a multitude of data, including various track marks, different types of data. In this model that I'm showing right here, they use these various epigenomic marks. And then you can use these to predict these non-coding elements, such as enhancers. And so you might be wondering, why am I showing a machine learning model from 2014? And the reason is because this was the first model that was used to determine whether HARs may function as enhancers. And so indeed, using this model that was trained to identify enhancers in the developing limb, brain, and heart, they identified that around 30% of HARs at that time were predicted to function as developmental enhancers in these various tissues. One thing that's interesting here is that you can see non-coding HARs are here in light gray, and here's a predicted fraction that are predicted to function as an enhancer either overall or in the various tissues. But the predicted function is similar to non-coding conserved regions. And conservation is something that's long been used as a predictor of functionality of the genome. The idea being that if it hasn't changed over millions of years of evolution, it's probably doing something important. And so with HARs, we know that those also were conserved in most species and are changing specifically in humans. So we know that they're doing something important and they're predicted to function as enhancers. And indeed, we can test these in vivo, and this is using a transgenic mouse assay. where they take the enhanced sequence, they attach it to a recorder again, and they look to see where it drives expression based on this blue staining. And so here you can see expression in the spinal cord, you can see expression in the brain and near the eye. If you zoom in, you can see that even more specifically. But using these types of assays, they were able to show that not only were these predicted to function as enhancers, but we could also show this in vivo. And interestingly, 30% of these HARs show differential expression between the human and chimp sequence. And so this is important because it shows that the sequence differences that happen in humans specifically are causing a functional change. So then we also looked at these in human neuroprogenitor cells. And so this was using what's called a massively parallel reporter assay, where you can take hundreds of thousands of sequences, attach them to reporters, and put them into a cell-based assay so you can, in a high throughput way, you can look at the function of these sequences. And so using these, we saw that around a third of these, again, were predicted to be active. The blue here in general is active, and then the dark blue specifically is human biased. And then here we're looking at human marks on the top and chimp marks on the bottom from neural progenitor cells. And we can see different patterns of activity over different stages of development. And so obviously this is really small and I know you can't really see it, but I've got the paper cited here if you want to take a look, it's very cool. But in this specific example, you can see for this HAR right here, which seems to be in the tab right here, that's human data. But then by comparing this human and chimp epigenomic data down here, you can see that there are some areas where they change. So for instance, later in development, this HAR appears to be more active in chimps than humans. And so how does that impact us over development is something that's still being investigated. But throughout this, you can see both how HARs have been tested in multiple environments, but also how technology has evolved. Because HARs were originally discovered in 2006, and obviously technology has evolved quite a bit. So we can go from testing one sequence at a time to a few to testing hundreds of thousands. And so one thing that's really cool right now is when HARs were first identified, it was using a sequence alignment of about 29 genomes. And since then, now we have an alignment of 241 mammals. And in addition, many of these genomes are much more complete than they originally were. So using this alignment from Xenomia, we wanted to identify an updated set of HARs. And so, as you all may know, in order to work with this kind of scale of data, you need to use higher computing resources. And so in order to facilitate identifying human accelerated regions in an alignment of hundreds of mammals, we implemented a pipeline with Nextflow, which is a nice way of managing a software pipeline, and it also enables you to deploy a pipeline on any cluster instance. And so using this, we start off by identifying, we use a species tree, a neutral model, and a multiple sequence alignment. With those, we identify things that are conserved in all species besides humans. And then you can then look within those elements to see which are accelerated in the lineage of interest. So in this case, we looked at humans and chimps, so identifying HARs and CHARs. But you can use this pipeline for any species of interest. So if you want to take a look at my GitHub, you can apply this to your favorite species. And so one of the first things we wanted to look at is whether these HARs look similar to HARs that have been found before, basically make sure our pipeline is working, we're finding the same things. And so we looked at the distribution of HARs, and you can see again, the height of this bar indicates how many elements fall in this category. So the majority of HARs are energetic. That's exactly what's found before. We find a lot that are intronic. We find a few more that are non-coding RNAs compared to before, and really that comes out to better indications over the 15 years or so that it's been since the HARs were first identified. We then used a pipeline to look at forces of selection acting on these HARs. And so this is not as straightforward as it is in the coding genome, where you can look at DNDS or patterns of specific exon usage. In the non-coding genome, you have to use different statistical methods that are looking at how fast this is evolving, and does this look like other things that could cause fast evolution, like GC bias gene conversion. And so by controlling for these factors, we found that the majority of HARs do appear to be under positive selection, indicating that they're doing something beneficial to humans and something that is a function that is being actively preserved. Some of them, of course, are just like gene conversion, as would be expected. And we see similar results for the chimp accelerated regions. So again, many ZooHARs are protected to function as neurodevelopmental enhancers. And so this is using an updated model that was introduced recently in a preprint in 2022. And this model is specifically looking for neurodevelopmental enhancers. And so we found that over half of HARs are predicted to function as neurodevelopmental enhancers. Here's an example of the type of data that goes into this kind of model. So you have thousands of data sets using things like DNA, Ataxi, different types of ChIP-seq. And you can see that the data types that are most predictive for neurodevelopmental enhancers are things that come from the brain, as you might expect. And then things that are less predictive come from tissues like the heart or the limb. So overall, HARs are still non-coding, they're still predictive functions enhancers even 15 years later. And so the question still remains, why are these loci evolving so fast in one species but conserved in most other mammals? And so it's been known for decades that the genome is meaningfully organized in 3D, but this has really caught on in the past 10 years or so. People have really started to investigate it using next-gen sequencing. And so we know that on a chromosome level, chromosomes tend to stick together within the nuclear space. If you zoom in a bit, the genome distinguishes itself into A and B compartments, where A tends to be more active and more on the interior of the nucleus, and then B is less active, heterochromatic, and associates with the Y fauna. And then if you zoom in even further, you can look at structures called TADs, or topological associated domains, and they either look like this, where this is distinguishing a part of the genome that likes to interact within itself more than with things that are on the other side of this boundary. And this also can look like this in a graphical image. Some technologies that have been developed in order to look at the 3D structure of the genome include protein capture sequencing. And so the way this works is that you use methods that essentially attach DNA to each other when it's close together in 3D space. And so even if something is far apart within the sequence of the genome, if it's folding near each other, it'll physically attach to each other. And then when you sequence through that DNA at the end of this experiment, you get these maps where the diagonal is the linear genome, and then these squares indicate how frequently that locus interacts with this locus. And the brighter the square, generally, the more frequently those loci interact. And then you get these patterns, example here, which is a boundary. So it's separating this bit of the genome from that bit, indicating that they don't interact as much. And you can also visualize this in loops like here. And again, similar to enhancers, we've seen that changes to the 3D genome can have major phenotypic impacts. And so in this paper, they were looking at various families that again have polydactyly, and they identified variants in a TAD that appeared to be associated with this. And what they noticed is that you have some genes here that have known roles in the development of your digits, and they identified that in one case there was a deletion that would impact a TAD boundary. There's an inversion and duplication over here, and some more over here that were leading to these different dactylies. And so then, by using H3K27 and ChIP-seq, they were able to identify regions within the central TAD that appear to be functioning as enhancers, and they validated these using transgenic mice, showing expression of the lymphoids. And then they were able to show the function of each of these structural variants within the context of causing these dactylies. And so, for example, the inversion was causing this enhancer to ectopically activate this gene. There you go. This duplication, again, caused ectopic activation of a gene by an enhancer. The deletion removed this boundary, and another deletion removed a separation between a boundary and a gene. So in all cases, it was rewiring this 3D structure to enable enhancers to contact genes they normally wouldn't. And this led to these disease phenotypes. So overall, the change in enhancer led to various dactylies, showing that the structure of the genome is important. And so we thought, in this case, this is a disease, but something similar might be going on in evolution. You get a structural variant that changes the 3D genome, you get a phenotype like this, and it's either positively or negatively selected against. And so one interesting thing that we found that suggested that something similar might be going on with HARs is we found that HARs were enriched with TADs that contain human-specific structural variants. And so specifically what we're looking at is, do HARs fall in TADs with human-specific structural variants more frequently than expected, based on a very rigorous null distribution composed of how frequently conserved elements, which remember are the broader set from which HARs are drawn, how frequently they fall in TADs with human-specific structural variants. And so again, just to remind you, so HARs are these fast evolving elements in the human genome, but they're drawn from a broader set of elements that are highly conserved throughout many other species. And so if we look at the statistical test, we have a null distribution where we're looking at the odds ratio of how often conserved elements fall into heads that also contain human-specific structural variants. And then if you look at that same, and this is over 1,000 permutations. And then if you look at that same value for HARs, you can see by eye that that's significant. So they're significantly enriched. So you've got these different human-specific forces occurring in the same 3D neighborhoods. And so this led us to ask, are human-specific structural variants changing the 3D genome around HARs or other species-specific accelerated regions? And so one way we wanted to look at this is we used a deep learning model that's able to predict 3D genome structure based on sequence. And so I know there's a lot going on in this figure, but what it basically comes down to is you input sequence, A's, C's, G's, and T's, and you output a high-C map. And so when this was validated, you can see the predictions here look very similar to actual validation data there. So this model is able to very accurately predict 3D genome structure based solely on sequence. And it's called a keto, which is very cute. So using this, we were able to look at what impact these structural variants might have on sequence. And so for instance, in this locus, here's the original sequence. And you can see that there's a strong boundary here. There's what looks like a cross-TAD contact here, a strong inter-TAD contact here, and a little bit of an inter-TAD boundary there. And then when you delete the specific human-specific structural variant, you lose this contact here. This contact is also lost, and this boundary gets weaker. So you can see that there's an impact there. And another way to compare these two types of 3D genome arrangements is by looking at the original and subtracting this one, so you can specifically see what's different. And so here you can see in the original, there's more context there, and a greater boundary there. And so, of course, these are still predictions. We wanted to validate them using high-C data. So we generated high-C data from human and chimpanzee neural progenitor cells, as you can see here, using the same methods I described earlier. And so using this, we were able to directly compare the 3D organization in neural progenitor cells of humans and chimpanzees. And so here again, you can see in humans, there's a boundary here, chimps, it's gone. Humans, there's some contacts here, those are missing in chimp. While overall in this locus, these are very similar. And actually a lot of what we learned is very new by looking at these data are that humans and chimps are very similar. And then here in the middle, this is a disruption score that summarizes the difference between the high-C of those regions. And so the high-C data validated 3D genome organization changes that were predicted due to human-specific structural variants. And so basically we were able to predict the impact using the human-specific structural variant and then go to the chimp data and see whether that matched our predictions, which suggests that those structural variants are what's causing the change. And so here in this locus, here is the predicted change. And so this is that subtraction map that I showed before, where you're specifically looking at the differences in the original compared to when the structural variant is removed. And so the difference is here, there's a stronger TAD boundary there and there's a contact there. And then the observed difference matrix, and so this is human versus chimp, you see the same thing. And so this supports the idea that this structural variant was causing the change that we see, and this was an evolutionary phenomenon. And so this particular locus is Zuhar 126. And then we can also look at human and chimp specific loops. And so these are significantly interacting regions in 3D. So for here, the pink is chimp and the blue is human. And then again, when we look at the human chimp data in this locus, it matches up with what was predicted. So again, we've got this TAD boundary here that's specific to human, and these contacts that are specific to human. And these appear to be caused by this human-specific insertion right here, which is very close to Zuhar 126. And so the idea based on this is that human-specific structural variants may have contributed to the acceleration of HARs and together may underlie human-specific traits. So I'm going to stop at this particular section, highlight co-authors in the study, Pavel and Sean, and my advisor Katie, who was very supportive of this work, as well as Xenomia, provided the alignment. And I'm going to transition to talk about epigenomics at Fauna Bio, where I currently work. And so at Fauna Bio, we are leveraging extraordinary genomics from the animal world for human health. And so as everyone here knows, we share 92% of our genes with rodents, and the sequence is 85% identical on average. Overall, there are a lot more things that are similar between us than are different. And so we're trying to leverage that for human therapeutics. And so here's an example from histone H1, where you can see that they're almost identical. There's a few positions where they vary. And we also know that animals are resistant to human disease pathologies. So for example, elephants very rarely get cancer. The naked mole rat seems to defy the laws of aging. And the 13-line ground squirrel is able to protect themselves from many diseases that humans would get if they were exposed to the same conditions they are when they hibernate. We are also trying to leverage hibernation induction for long-range space travel. So for example, conserving resources during long voyages, protecting bone and muscle from the effects of microgravity, minimizing psychological stress, and minimizing effects of radiation. And so one species that we've been focusing on is the 13-line ground squirrel. And so this is a body temperature trace of a squirrel going through hibernation in the winter. And so you can see when the squirrel is awake, it has a relatively high body temperature. And then it goes into these periods of very low body temperature with interbout arousals interspersed, where the squirrel comes back up to temperature and then goes back into torpor. And so here's an interbout arousal zoomed in. And so this is the equivalent of experiencing 25 heart attacks per year. If that happened to us, we would die. So we're trying to figure out how squirrels are able to manage this amazing physiology. And so one thing that's really cool that happens during hibernation is that during the entrance to torpor, the neurons in the hypothalamus, they become hyperphosphorylated. Their tau becomes hyperphosphorylated, which is a physiology that's associated with Alzheimer's, frontotemporal dementia, and various other diseases. And then during arousal in the IVA, this goes away. And so if we can mimic this, potentially this could be therapeutic. Another thing that's really interesting about these squirrels is that they are protected, their hearts are protected from damage. And so here on the left, we have a rat that's exposed to ischemia reperfusion, which is basically what happens during torpor. And you can see in the top, this is a healthy tissue. This is what it's supposed to look like. And then after exposure to these conditions, the tissue is quite damaged. It has a lot of leukocyte infiltration. And then here we're looking at a squirrel in the summer as well as in the winter. And you can see between the control tissue and the tissue exposed to ischemia reperfusion, there's a little bit of damage in the summer, but there's very little in the winter. And so figuring out how squirrels are able to do this could be extremely therapeutically relevant for humans. And so together, Fauna is based on multimodal data that enables precise target discovery. This includes hundreds of animals, which is increasing every day, including species. We don't just work on squirrels, we work on any extraordinary animal. We work on various tissues, various time points. We're working with Genome, which I discussed earlier, to study all these in context, and various types of data, including epigenomics. And so our core process takes this data. We use a lot of network biology to figure out the connections between these nodes. Using WGCNA and other methods, and integrating human data, as well as disease-specific curated data, we identify targets which are often network hubs or high-ranking modules, and those become therapeutic targets for downstream development. But as we all know here, the answers to many evolutionary questions lie in the non-coding genome. It's not all just genes. And so I showed the example earlier with enhancers contributing to polydactyly. There's also examples of this same enhancer contributing to the loss of limbs in snakes, so an evolutionary process involving the same enhancer. And so what I'm showing here is here's a multiple alignment of various snakes. The basal snakes are the ones here that still have vestigial hind limbs. And what's really cool here is that these basal snakes, their ZRS enhancer is more conserved with other species that still have limbs. Whereas what are called the advanced snakes here, they have no more limbs. And in some cases, they've completely lost this enhancer. In other cases, it's degraded to the point where it's no longer functional. And so just to show an example of how the non-coding genome participates in both disease, but also evolutionary processes. And so at Fauna, we want to leverage this. And also, regulatory elements may explain many outstanding questions in hibernation biology. So looking at these super cute squirrels, one of our founders, when she was still an academic, she did a study where she was looking at genetic variants that significantly associate with the onset of torpor. So you can see in this squirrel, it goes into torpor a bit slower, takes a little longer than this squirrel, which is more abrupt entrance into torpor. And so she identified variants that are associated with these variations. But we know that most GWAS variants fall in the non-coding genome, making them difficult to interpret. And so this is shown very effectively right here, where the majority of GWAS variants are intronic or energetic. Excuse me. And so the same was true for this torpor induction GWAS. And so through some follow-up work, she showed that some of these variants do have regulatory activities. So for example, one of them falls in just upstream of the 5' UTR, probably a promoter region, which is also conserved. And it interacts with this transcription factor binding motif and changes its impact on the target gene. And then there were also various other SNPs from this GWAS that function as eQTLs indicating regulatory activity. And so by digging more into these, we can get a better idea of how the regulatory genome is participating in hibernation regulation. And this broadens our search space for therapeutic targets. And another thing that's very important for therapeutic development is that sometimes modulating a gene for the whole body has detrimental effects, but modulating that same gene in a very specific tissue can be therapeutically beneficial. And one way to do this is with regulatory elements, which can be cell type specific. And so here's an example of a regulatory element. On the top, this says cell type specific function. In this case, it's mediated by cell type specific transcription factors. So when those are in a cell type where they're expressed, that regulatory element is activated. When they're not expressed, it's not, as compared to other regulatory elements which are more global. And indeed, cutting-edge genomic modulation technologies enable targeting of these regulatory elements. For example, CRISPR inhibition, which is a deactivated form of CRISPR, which is able to, you put a inhibitory domain on deactivated CRISPR, so it doesn't cut, but it just attaches to a specific region and then deactivates it. You can modulate regulatory elements that way, and then decrease expression of their target gene. And the opposite with CRISPR activation, where you can turn on specific genes via the regulatory elements. And so all this to say that Fauna Bio is not only not restricting ourselves to human biology, we're looking at the entire animal kingdom, but we're also leveraging evolutionary innovations throughout the whole genome to treat human disease, not just genes. And here's the team, and I'll stop here.
Kathleen, I had a question that I've been thinking about for a while about the first part of your talk. So I've been hearing about the work for a while. I know that you did some investigation as to what genes are in these tags, these boundaries are seem to be changing all the time. I was wondering if you, based on what you know about those genes, have any hypotheses as to what human-specific traits, so traits that differentiate humans from other mammals, might be influenced by tags.
Yeah, so we see enrichment for genes that are involved in neurodevelopmental processes, especially genes that contribute to brain size, which we know is a human-specific phenotype. And so we think one of the most likely things that may have happened is the expansion of the cortex seems to be a likely candidate. And I say that with a little bit of hesitation because these also suffer from bias because those genes are the most studied. So I think that's definitely part of it. But I don't want to take anything else off the table. No. Yes.
So when you're comparing the IC data with chimp with human, there must be the 3D contact difference between the species, but there must be also one-dimensional difference because their genome learning from the chimp as well, right? Mm-hmm. So it must be very difficult distinguishing the two from the IC data.
Yeah.
Can you explain a little bit more about that?
Yeah, so the way we handle that, you're right, that would be very difficult. And so initially we try to compare them using their respective reference genomes. And there's a few reasons why that's difficult. Like you said, there's a lot of rearrangements, but also the chimp genome is much lower quality than the human genome. And so what we actually ended up doing is mapping everything to the human genome and comparing it that way. And so there are obviously limitations to that. We're missing a lot of the rearrangements and stuff. And so we focused on syntenic regions and only compared those. Yes.
I don't know if you touched on this or not, but what do you think actually maybe causes the increase? Because you said that they weren't very conserved. Do you guys have any ideas maybe what's caused like you're going into what those repercussions might be, but what's actually causing the increase in the lack of concentration in the HARS components. Does that make sense?
Yeah, that makes complete sense. That's actually something that was driving this work, because I was thinking about this, you know, and I was like, HARS are very cool. Obviously, we think they're causing human specific things, but why? Why do they accelerate humans? And so the idea, and this hasn't been proven yet, and I think it's going to take years to try and figure this out, but the idea that I have is that the changes in the 3D environment around HARS put them in contact with a new gene, and that different contact caused a small change in a phenotype in humans that may have been advantageous, and that therefore having more of that would have caused the heart to change, maybe in a way that caused it to activate more.
Like a cascading?
Yeah, cascading effect. But another thing that's been shown since then, I think that's one thing behind it, but also it's been shown that HARS tend to sort of buffer each other. And so a change in one direction could cause a change in another HARS to sort of buffer that change, so nothing too drastic happens at once. So it seems to be a concerted evolutionary process. But it's not completely understood yet, and I think it's going to take a long time to figure that out.
Great, so I think right now, what we have available to us is some time for, we're here, we go back, we're going to be eating lunch at noon. But it's an opportunity to go and talk to Kathleen and Jason, ask them specific questions you may not have wanted to ask them in the room. Teachers, Jen told you earlier, you're more than welcome to go and come to somebody like me or these guys and ask them something that makes it a little bit more accessible for you, because I know that some of this wasn't not accessible. So we can sort of interpret, if you will. Then we're going to break up and go into the groups after lunch. That sound good? Awesome.